YouTube.
Yeah, I gotta figure out that that's a good good use of technology.
Go YouTube. Okay, we are on YouTube live streaming now.
Awesome.
Nunu, you can start.
Okay. Yes. I think still people are still joining the Zoom. But let me start introducing our today's speaker, Professor Taisino. So Professor Taisino received his bachelor degree in both chemical engineering and in chemistry from University of Pennsylvania. Then he received his PhD degree in chemical engineering from MIT. After that, he moved to University of Pennsylvania Department of Chemical and Biomolecular Engineering as a faculty member since 1999, and he is currently a professor of the Chemical and Biomolecular Engineering. He also has the second appointment in the Department of Mechanical Engineering and Applied Mechanics at Penn here. Also, Professor Taisino also serves as the founding director of the master degree in scientific computing at Penn. His current research in computational materials and is focused on multi-scale modeling and simulations of nucleation, assembly, and crystallization processes in a wide range of materials systems. Examples include semiconductor microstructure evolution during crystal growth and wafer processing, colloidal self-assembly, and platelet cell aggregation in blood flow. His group develops and applies numerical computational techniques across length and time scales to study these problems, including molecular dynamics, various flavors of molecular and continuum modeling. So today, Professor Taisino will share his talk on non-equilibrium phenomena in the self-assembly of DNA-linked colloidal crystal. With this, welcome Taisino, and the floor is yours.
Okay, thanks very much, Lou, and thanks also for the invitation. So let me reshare my screen here. Where did it go? There we go. And there we go. Yeah, so thanks again, and thanks for everybody for attending. So I'm going to tell you a little bit about our work. This is kind of a long-standing project on looking at self-assembly. It's mostly mediated, or I should say, motivated by DNA-linked colloidal crystals, but a lot of the phenomena I'll talk about today are somewhat generic, and I think they're general features of a lot of colloidal systems. So before I start, let me just make sure I squeeze in a couple of acknowledgements. So some of the former students and postdocs I've had working on this include Ronaldo Scarlett, who did some of the very earlier work, Ian Jenkins, and most recently Young-Kee Lee, who was a postdoc in my group and is now a faculty member back in Korea. And then I'd also like to acknowledge collaborators along the way. So John Crocker, one of my colleagues here at Penn in the chemical engineering department, whose experiments really motivated most of this work. And then more recently, Professor Celia Reina and Paris Perdicaris, both of whom who are in the mechanical engineering department at Penn, who were involved in different facets. I'll mention that as I get along. This work's been supported by NSF. So at this point, I think DNA mediated particle assembly has been out long enough that perhaps many people are familiar with it. But just very, very briefly, and let me, I always forget how to do the pointer here. Let me see. Where is the pointer? Lou or Jen, do you guys know where I can find the pointer on this?
Yeah, so on top of the Zoom, you can maybe you can use the annotate function.
Oh, yeah. I guess I can do that. So I'll try Spotlight. Here we go. Can you see this?
Oh, yes, it's great.
Okay. Great. Okay, so so the basic idea is you've got some particle, and then you've got some chemical way of attaching single-stranded DNAs to these particles. And these are designed in such a way that a certain piece of the DNA strand is complementary with another piece of that DNA strand on a different particle. And the kinetics of this binding or hybridization and unbinding is rapid enough to give you some kind of a time averaged interaction between these particles. And so as the particles come closer to each other, more of these bridges can form, and you create a stronger bond. As they get pulled away, less bridges form, and then that weakens the interaction. And then as you really bring them close to each other, the polymer brushes, the DNA strands start repelling each other entropically. And in a sense, you end up with a pretty familiar, if you do molecular simulations, a kind of a time averaged interatomic or interparticle potential pairwise that looks something like this. It's repulsive at short range. It's attractive somewhere in the middle, and then it goes to zero. And this is kind of well understood, especially when we're looking at the situations I'm going to be telling you about, where the particles are rather large and the DNA strands, rather large, I mean on the order of a micron, and the DNA strands are rather small, on the order of, let's say, 50, 60 nanometers. And so you can think of these kind of approaching the limits of sticky spheres. And certainly the interactions between a pair of particles is not affected by a third particle, let's say out here. So the interactions are very much pairwise. We can model this quite well. And the experimentalists can also measure this. Oh, that's not what I wanted to do. Let's go back. Okay. Just trying to get rid of this this thing here. Okay, doesn't matter. So these are examples of the potential, the pair potential between a pair of these particles, and they're color coded according to the sequence that is, to the DNA sequence that is on each particle. So red particles have this sticky bit, and blue particles have this. And what's interesting about this is that this is not self-complementary. So if you brought another blue particle and tried to put it up here, it wouldn't match with this one. So it's selective. So red always meets with blue, but blue doesn't bind to blue and red doesn't bind to red. So in this case, if we label red and blue as A and B, there's interactions between A and B, attractive interactions between A and B, and they would look like this, but there's no attraction between A, and you'd only see the repulsive part, okay? The other subtle detail here is that the extent of hybridization at a fixed separation between these two particles is very much temperature dependent. And so you can go from having rather strong attraction to weak attraction over a rather small temperature range. Okay, so this is built into the potential that we feed our simulations. Okay, so we take this information, we pass it into either a molecular dynamics simulation or a Brownian dynamics simulation or even a Monte Carlo simulation, and we use this to run things. And before I get into the simulations, I just kind of want to give a very brief history of the field from an experimental perspective. And I say brief, it's brief and selective. So what that means is, or I should say, this kind of two parallel paths. There's people who've been working really at the nanometer scale with nanoparticles. And these particles are typically in the 10 to 20 to 30 nanometers in size. And then there are people who've been working at the micron scale, where particles are typically on the order of half a micron and up. And you know, the basic idea is the same. The difference is that at the nanometer scale, the size of the DNA strands that's put on the surfaces is significant compared to the particle size. And the other big difference is that it's pretty hard to generally see and image the structures that you get at the nanometer scale in general. And so it's much easier to do this at the micron scale, where you can use optical microscopy to image structures that you get. And very loosely speaking, so this was, you know, back in the late 90s, this was proposed both by Merkin and Oliver Satos's groups as a way to drive self-assembly. And the initial efforts really led to just aggregates. It was very difficult to control how the assembly proceeded in order to form organized structures. And then over time, this led to making crystals out of various combinations of particles. And then again, Merkin's group in 2011 really demonstrated that you could do this with a whole variety of different particle size ratios and different compositions, so that you could make a wide host of different crystal structures. And then, you know, most recently, people have been working on kind of more evolved aspects of this, looking at crystal structures that are transformable. And I'll talk quite a bit about this because it's one of the phenomena that we decided to look at. On the micron scale, I think generally speaking, fewer demonstrations of a wide variety of structures have been put forward. And in part, this is kind of a statistical thermodynamical issue, which is that you're closer to having a bunch of sticky spheres, and sticky spheres are less easy to equilibrate. And two, I think the chemistry in terms of what you can, you know, how many different particle populations you can label, etc., is a little bit smaller. And so there's been fewer different kinds of structures being demonstrated. But the nice thing, like I said, is very recently the ability to pattern how you put things on particles and even creating clusters of particles has been developed. And so now people are being able to make some really nice high void space structures that are optically interesting. I'm not going to talk too much about that particular aspect today, just for time's sake. So just to go back to the system that, so I want to kind of take you back a little bit to some of the earlier stuff, because it provides a foundation to the things that I will talk about later on. But this is the system that we're interested in. I've got particles of type A, particles of type B. Again, these are just strictly They're the same particles, just functionalized with different partial sequences of these single DNA strands. Okay, and in this case, the red strands are only complementary to the blue strands. Okay, so red and blue bind together, red and red don't, blue and blue don't. But what we can do is we can mix a little bit of red strands with the blue strands and have systems like this, where if I have a mostly blue particle, it will interact with another mostly blue particle through the red-blue interactions. But this is a weaker interaction. We can tune it by changing the mixture of red and blue. Okay, so, and if I have equal amounts of red and blue on every particle, then I effectively have a single component system. So we can go all the way from a single component system to a purely AB system by changing the ratio of the red and blue strands on the particles. Okay, so that's kind of the universe of interactions we're going to really look at today. And while this is a pretty simple system, it's already pretty rich in terms of a lot of phenomena. So it's plenty to understand the basic ideas. And this is just a demonstration. Okay, so this thing is really irritating me in terms of position. Here we go. All right, I'm going to put this down here. Okay, so this is just an optical, a confocal image of a crystal that was grown in John Crocker's lab. And just to demonstrate that you can unequivocally see that this is indeed a crystal structure, you can see just about, in this case, the individual beads, the three-dimensional nature of the crystal, and also the facets. And these are essentially, the way this is done is the particles are put into solution, and then the temperature is gradually lowered in time until the interactions become strong enough relative to the temperature, to the thermal energy. And at some point, the particles start homogeneously nucleating into little crystallites. The crystallites then continue to grow. And then eventually, while each particle is pretty density matched so that it floats in solution, eventually these crystals become large enough that this very slight density mismatch means that these crystallites fall out of solution to the bottom of the vial, at which point they stop growing. So if we look at structure, and there's really not a whole lot of crystallography that we're going to do here. I'm just going to mention two basic structures that we're going to think about. If I just have AB interactions, okay, so this is either red or blue particles with no mixing, and with a size ratio where the A particles are slightly smaller, about 15% smaller than the B particles, the structure that you see, and in this image, in this confocal image, you can see the surface of one of the crystallites that forms, and the particle, the A and B particles, are labeled by red and green fluorescent entities. And so that way we can actually see the structure of the binary crystallite that we form. And in this case, the binary crystallite that we form is an analog of cesium chloride, which is a BCC, or a body-centered cubic lattice with alternating red and green particles. Okay, and this is what forms. And the main thing you need to keep in mind for now is that each A particle sees 8 B particles, is bonded to 8 B particles, and each B particle is bonded to 8 A particles. Okay, and that is the structure that you see under these conditions. If you change things a little bit, so now we're going to a size ratio that's closer to 1 to 1. And instead of having a pure AB system, we have a system where some AA interactions and some BB interactions are allowed by mixing some of the A and B strands on the respective particles. And you let that system evolve. Now you get a very different configuration. and I'll refer to this as, so this is the atomic analog to this is copper gold. So we had cesium chloride, now we have copper gold. Copper gold is an FCC, and again, I'm alternating red and green, okay? And an FCC, which is a close-packed 12-fold coordinated system, each A particles will have bonds to 8 Bs and 4 As. And each B particles likewise will have bonds to 8 As and 4 other Bs, okay? So, and very simply to understand this, the extra AA and BB interactions offered by this mixing is what favors this structure. It's more densely packed, which is entropically not favorable, but it's enthalpically favored by the additional interactions that we have. Okay, and so that's why this forms under these conditions. So now we can take a little sidestep. I just want to, I don't want to spend too much time on this, but I just want to point out that when these crystallites are growing, this is just a picture from a simulation, particles that are landing at the surface can land in the right place, or they can land in the wrong place, in which case they form a defect. And the defects here that we're talking about would be having an A particle land in a site that's meant for a B atom, or a B particle landing on a site that's meant for an A atom. And these are known as antisite defects. So you can think that there's an energy penalty for forming such a defect, and there is. And we can calculate what the energy penalty is on the surface of a FCC, or a close-packed crystal. And I can do the same calculation on the surface of a BCC, or a cesium chloride crystal. And it turns out that the cost of forming an antisite defect is higher on the cesium chloride, or on the BCC, than it is on the close-packed crystal. And what does that mean? It just means that thermodynamically, you'd expect the cesium chloride to grow more ordered, because the cost of forming a defect is higher. And the lower the cost of forming a defect, the more defects you will have as this thing grows. So now from this, and you can ignore all of these equations. So this is just like an isotherm. This is the cost of the antisite. Okay, all of these units are always in kTs. Okay, so this is kT, 4 kT, 6 kT, and so on. And this is the, you can think of this as an order parameter where 4 is perfect and 3 is random. Okay, so this is a lot of antisites. This is no antisites. And you can see that the isotherm for the BCC case quickly reaches perfect ordering as the cost of the antisite goes up. Whereas in the close-packed case, you have a lot more tolerance to defects based on a given antisite energy. Okay, so the reason I mentioned this is just to point out that if you take this into account, you have to kind of think about the free energy of these relative crystal structures, not from a perfect crystal structure, but from the crystal structure you can actually make at equilibrium. Equilibrium says I'm going to have a finite number of defects, and how many defects I'm going to have depends on the cost of those defects. If they cost little, I will have a lot of them, and so on. So this is a phase diagram for what you would expect to see as this is the interaction between A and B. Think of this as the dominant interaction. And then this is the interaction between like particles. So either AA or BB, doesn't matter. So when the interactions is low, you don't get any assembly. As you crank up the interaction, you form this BCC, or cesium chloride phase. And then at a given interaction between A and B, as I move this way, I'm increasing the attraction between like particles. I cross this line and I enter now the close-packed equilibrium phase. These lines represent the ideal ordering, or the ordering extent. So if I have a lot of, if I come this way, or I'm up here, I'm going to have high degree of ordering. If I come down here, I'm going to have a lower degree of ordering. And by the time I get to this line, it's still close packed, but I'm going to have a random mixture of A's and B's in the system. So, so this kind of, this is purely based on thermodynamic analysis. This is assuming that I can grow this phase and this phase at equilibrium. But in fact, nucleation and growth are non-equilibrium phenomena. And so I can't really achieve these. I can only achieve these if I'm at equilibrium. So this is the same figure. I just want to go back to here. This is the same figure as this. This is the isotherm based on equilibrium. This is what you actually see based on running simulations. And each of these points, what it's telling you is that, so the hollow points, the hollow symbols represent close packed crystals. The solid symbols represent BCC. So what you see is all the BCC cases always lie up here. So they're always perfectly ordered. And all the close packed crystals lie below the isotherm. That says that I'm being affected by kinetics, because the amount of time, so just to kind of give you a simple picture, if I have a crystallite and I have a particle attached in the wrong place, it can leave the wrong place and try again and come back to the right place. If I have an infinite amount of time to try out wrong and right places, I'm under equilibrium conditions. But since this thing is actually growing, I only have a finite amount of time to correct mistakes before I'm buried by another layer of particles. And so that's why there's a kinetic limitation. And the cases where I am very far from what equilibrium predicts are cases that correspond to rapid growth. Okay, so you can think of it as you move away from this line downwards, you are being subjected to more and more kinetic limitations. What this basically means is that the close packed configuration is never as favorable as you would think it would be, because it's subject to this very defective growth. I'm going to skip this for now, and I'm just going to go to here. So this is now a phase diagram that was generated experimentally in John Crocker's lab. And here's the big picture. So this is the same thing, AB interactions this way, AA interactions this way. I've got close packed, I should say, cesium chloride crystals in here. I've got close packed crystals in here. These lines represent different amounts of mixing of this red and green strands that I was telling you about. So as I mix more, I follow lines that bend this way. Okay. Now, if you go back to this picture, you can imagine that if I go this way, I'm going to be less ordered than if I go more vertically. And what they ended up doing was counting every crystallite that they saw. So this is a plot of the statistics of what they see. So as I go from this line, so this is an alpha of 0.06, that means I have 6% mixing. So 0.06, most of the crystallites I see are cesium chloride, and I see a few of these copper gold. As I go to 0.1, which is this one, most of the crystallites are cesium chloride, or I should say fewer crystallites are cesium chloride, and more of them are copper gold. And then I'll keep going. By the time I get to 0.16, 0.16 is this, almost all of them are copper gold, very few are cesium chloride, and some of them are this orange color. So FCC orange means that I have an FCC crystal that is compositionally disordered. Okay, and so if you compare this picture to this picture, you'll see these have both the same structure, but the red and greens are randomly ordered here, and they're perfectly ordered here. If you run simulations, we can get roughly this picture, but the problem is we never see this perfectly ordered copper gold because of what I just told you, because of defects. Defects kill perfect ordering, yet the experiments show perfect ordering. And so this was kind of a mystery, and it's kind of one of the topics that I'm going to talk about today. And it turns out that these copper gold crystallites don't actually nucleate homogeneously. What happens is that crystallites grow cesium chloride and then undergo a martensitic transformation to copper gold. So when you follow one of these lines, as you're in here, you form a cesium chloride crystal. And then by the time you push it into this part, it will spontaneously just change shape and form a copper gold crystal. And the evidence for this was pretty profound. If you look at some of these crystallites, you can actually see crystallites that have half and half character. So you can see that this was a crystal that started as cesium chloride, and then it transformed into copper gold. And this was caught in the middle of that. This transformation was arrested in this case. And we see many of these. And so this was a very interesting thing, because martensitic transformations are very useful and well known in metals. So shape memory metals like nitinol, which is technologically very useful, goes between a BCC and an FCC configuration upon heating and cooling, again, driven by entropy and enthalpy importance. So importantly, these are often referred to as diffusionless transformations. These are literally, there's no long-range diffusion. It is just simply shearing or deforming the system in some way that allows this to happen. So we got obviously interested in this as simulation people. And by the way, I just want to show this picture. Subsequently, John Crocker's group actually went through and tried a whole bunch of different size ratios and different stoichiometries and showed that this happens quite ubiquitously. It happens quite generally. And they were able to, so this is the copper gold here, but they were able to make a whole bunch of different configurations that clearly came not from a one-step nucleation process, but rather nucleation of a parent phase followed by transformation of a child phase. So this happens and others now have seen this in several other systems. And so one starts to think, maybe we can use these as kind of, transformer crystals that can undergo different phases depending on the ambient conditions. So that's why we got interested in studying this. So just to give you an idea, this is cesium chloride, which is the BCC configuration. If you take the 110 planes in BCC and you just shear them, you go to FCC. That's a very simple pathway. And this is a well-established martensitic transformation pathway, the Nishiyama-Wasserman pathway. If you do a stretch, a uniaxial stretch, and then compression in the other two directions, this is called the well-known bain pathway, also goes from BCC to FCC. And then there are other pathways. I'm going to skip this. There are other pathways that if you take each plane and you slide them up and down in an alternating fashion, you go from BCC not to FCC, but you go to HCP. Okay, so it's another ordered phase. It's still close packed, and it's degenerate energetically with copper gold. And so that says that as I slide planes up and down, I can either form HCP, I can form FCC, or if I mix the two, I can form random HCP. Okay, and all of these have the same energies. So we started doing some simulations and watching crystallites undergo these transformations. And what we saw was, as you would expect from a kind of a simple statistical analysis, that you would see both HCP and FCC in a random way. And so the color here, red is HCP, green is FCC, and indeed they're random. So whenever you start out with BCC, you cut the crystal in half just to view it, and you'll sometimes see this, you'll sometimes see this, sometimes you'll see transformation starting in this direction, then in this direction, and then they meet here and the crystal gets locked and it can't finish. So we started asking, well, why do we see almost exclusive FCC in the experiments, but we see a mixture in simulation? So we started varying different things. We were doing Brownian dynamics. We varied the diffusivity through the viscosity. We still see a mixture. We varied the crystallite size. Small crystallites, large crystallites, again, it's a mixture. So crystallite size doesn't affect anything. The viscosity doesn't affect anything. We varied the relative interaction strengths. Doesn't affect everything. Doesn't affect anything. And as you can see, most of these, although it's random, it's mostly red, which means it's mostly HCP. It's about 80% HCP, 20% FCC. So the only thing that was left was either our model's wrong, in other words, the interaction potential is completely wrong and somehow that changes things, or something else is going on. And of course, long story short, something else is going on, which is the one thing we weren't thinking about, which is hydrodynamics. And so we started investigating the use of this method, multi-particle collision dynamics or stochastic rotation dynamics. I don't have time to get into this. I'm running late, so I'm just going to basically say this explicitly accounts for the hydrodynamic interactions of the solvent in which these particles are floating around. And so the idea is that if a particle moves here, it's going to disturb the fluid, and then that disturbance is going to then be transmitted to this particle, which will then be dragged with that fluid. And so these hydrodynamic interactions could then bias the system. So, and indeed, we took our base case Brownian dynamic simulations. We started, so this is the percentage of FCC that you see. And like I said, it's roughly 80-20 without hydrodynamics. We went to MPCD and we saw that now we went from 20% FCC to 60% FCC, which is still not 100%. And after some more thought, we started realizing that MPCD doesn't capture short-range interactions very well. There's a lot of technical reasons for that. It's a problem we're actually still working on. As we introduced some approximation for the lubrication forces between the particles, we got to closer and closer to 80%. So it's clear hydrodynamics does bias this process. So let me show you just a couple of quick movies. These are movies. So this movie here is a set of the instantaneous velocity vectors for each of the particles in a crystallite. Okay, and this crystallite is undergoing a transformation from cesium chloride, from BCC to FCC. And you can barely see anything in here. You can kind of tell that at the beginning, the lines are a little bit longer than later. That's the only signature that you have of the transformation. And this movie is the same exact data, but now we're colorizing everything so that you can see that indeed I go from cesium chloride, which is blue, to red and green, which is HCP and FCC. So this is without hydrodynamics. If you then go and include MPCD, now you can see that the motion, the velocity vectors, are a lot more correlated with each other. And early on, you can see that you can start to see these swirl patterns of the particle motions. And that's because hydrodynamics is correlating the motions of the particles. And then once it transforms, you see that the transformation leads to a significantly larger number of FCC particles than in the first case. And when you add short-range forces, and I'm going to emphasize these are only approximate short-range forces, you see something even more correlated. So now they become really correlated. The transformation becomes very clear, and I'll see if I can find, yeah, so it's literally right in here. The instantaneous velocity vectors become very organized. And so this whole deformation is now taking place in a solvent controlled way. And then once it's over, then I just have small vibrations. And again, if you look at this, the particles really move much more subtly now because they're constrained by their hydrodynamic interactions. And once the transformation takes place, you get an almost complete FCC crystal, which is a lot more consistent with the experimental observations. So let me just briefly walk you through what we tried to do here. So we wanted to study this transformation pathway. And if you do a mode analysis on these crystals, you find that there's a whole bunch of zero modes. Zero modes means I can twist or deform my crystal along certain eigenvectors and at zero energy penalty. So in other words, these crystals are floppy. And this is what allows them to transform from cesium chloride to close packed. So there's about, in this crystallite, which was, let's say it's about 1,000 particles. So that's why I have 3,000 different eigenvectors, of which about 75 are zero modes. So we took these eigenvectors and we tried to follow the crystal deformation along these eigenvectors. The problem is these are all degenerate. They're all zero frequency modes. And if you look at these zero frequency modes and you try to follow them, you see this random noise because they're all linear combinations of things. There's no way to extract out clean dynamics because of this degeneracy. So what we did is, this is when we started working with Celia and Paris. We did a principal component analysis, and we tried to find an automatic way of ordering these modes. So instead of trying to separate them based on frequency, we tried to separate them based on variance. And now when you try to separate them based on their extent or their variance, now it's no longer degenerate. So this is, again, all 3,000 modes, or I guess this is a slightly smaller crystal. That was 1,100 modes. You can see up here, there's 6 very large amplitude modes that correspond to translation and rotation. And then you can start looking at these large variance modes, and you see that, in fact, they do correspond to very well organized transformation pathways. This is like the bain transformation. This is some combination of bain in 2 directions, and so on. And now we can take all of these well-organized or relatively well-organized modes, and now we can ask, how does the system evolve along each of these modes? So what we did is we took the trajectories and we projected the trajectories along each of these modes. And if you do this, you can compute diffusivity along each of these directions. And if you do this with Brownian dynamics, what you find is there's 2 types of trajectories. There's trajectories that have high diffusivity, and then there's trajectories that have low diffusivity. But all the high diffusivity trajectories have the same slope at early time. When you do this with fluid mechanics and included, now you get a much broader dispersion of diffusivities. You get high diffusivities, medium diffusivities, low diffusivities. It's a lot more dispersed. And if you look then, what is a high diffusivity versus a low diffusivity pathway, you can now plot all the diffusivities along these different variances, along the different eigenvectors. So in BD, all the different modes, so the x-axis is just modes, I'm just using variance as a plot, as an axis, all the diffusivities are the same. When you have fluid mechanics, the diffusivities now vary. And in fact, what's interesting is that they're actually higher than the Brownian dynamics diffusivities. And that's because fluid mechanics actually assists correlations. And that allows things to slide over each other more easily because the motions are correlated. And when you add lubrication, that starts creating shear friction. And so what that does is lower all the diffusivities. But the important point is I now have a very wide range of diffusivities along different eigenvectors. And long story short, the high diffusivities correspond to FCC generating outcomes, and the low diffusivities correspond to HCP generating outcomes. This is how fluid mechanics biases the outcomes of these transformations. I'm going to skip this. I don't know how much time I have. Do I have another 5 or 6 minutes?
Yes, you have it.
Okay, great. So let me skip this little bit and just briefly mention a second phenomenon that I wanted to talk about. So, all right, so one of the problems with all of this, and I mentioned this at the beginning, is because of this very strong temperature dependence, these crystallites are pretty hard to form. You've got to cool your sample really slowly, otherwise you'll form a gel. And so there's a tiny window for crystallization. If you stay at high temperature, there's no aggregation. If you go to low temperature, you form a gel. And there's a tiny little window where you give the particles enough time to rearrange and form a crystal. And this is particularly bad when you use large particles, micron scale particles. Not such a big problem for nanoparticles, but definitely a big problem for micron scale particles. So we, and I guess I don't have time to tell you how we started thinking about this, but one thing that I didn't mention is that when you make these particles, you're basically putting them in a solution filled with these DNA strands, and then the DNA strands join or become bonded to the surface. The number of strands that bond to each particle is not exactly the same for every particle. There's a heterogeneity there. And particles that have more strands will bind more strongly to other particles, and particles that have fewer strands will bind less strongly. And so instead of every pairwise interaction being the same, there's a distribution of interactions. Each particle has this factor B that says I either have more or less than the average number of strands on me, and therefore a pairwise interaction then becomes Bi times Bj times the mean interaction strength. So this is something we call interaction heterogeneity, and we're very interested in this effect because it seems to have a profound impact on self-assembly. So imagine now that I have a bunch of particles and there's a Gaussian distribution of binding strengths between the particles. And this distribution might have, a mean of 1 and a sigma of 0.1. Okay, so that's a typical amount. And this sigma, this standard deviation, or I call it P here, depends on the chemistry. Okay, but for a lot of the chemistries that people use, it's typically in the area of 0.1. So given the limitation on time, I'm just going to go straight to this. So this is 3 sets of simulations at 3 different interaction strengths. Okay, so in this case, what I'm showing you are only the particles that are bonded to other particles as part of clusters. So what you see is if I have an interaction strength, and these numbers don't mean anything, but this is a binding strength of 4.8, it's subcritical, which means most particles aren't clustered. When I increase it slightly to 5.2, all of a sudden I form these big crystals. When I go to 5.8, the whole system just gels up and I form these kind of crystalline gels. So there's a pretty narrow window of going from nothing to gel. And there's only a tiny window in which I form nice big crystallites. So this is the standard case where every particle is the same. Now, when I go to an interaction heterogeneity of 15%, so now the colors on the particles represent whether they're strong or weak binders. So red is a strong binder, blue is a weak binder, and green is an average binder. And now we go from same range. So in this case, from 4.8 to 5.8, but this time I go from 3.6 to 4.6. What you see is that you maintain crystallinity in the system over the entire window of this one kT of interaction strength. And it's because having the heterogeneity actually stops the system from gelling. It's kind of an inhibition of gelation. And the reason for that is because the strong binders recruit particles first, while the weak binders are still not yet driven to aggregate. And so instead of having nucleation take place all at once, it kind of spreads this nucleation process out. And so this is kind of another situation in which when I have a non-equilibrium situation where I'm growing crystals, having a real world effect. In the first case, it was hydrodynamics. In this case, it is heterogeneity across particles. I inhibit gelation and I make crystallization more robust. And so I'm going to, in the interest of time, maybe just show you one last thing, which is this is a plot of time, and this is kind of something we didn't really clean this up, but I'll just basically mention the following. So this is time. This is the number of clusters formed in the system. And this cyan here is the number of clusters that form as we cool a sample. So this is a non-equilibrium simulation. So you see nothing, 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 nothing. And as we're cooling, all of a sudden the system is triggered to aggregate. And then the cluster count goes up. And then it starts coming back down because clusters are joining, they're coalescing with each other and creating this gel. So if I include now, instead of having all the particles have the same interaction strength, I start putting some buffer particles. I'm going to add some subset of particles that are stronger binder particles. What happens is that I drive this process to happen at an earlier point in time, because now these can aggregate at higher temperature. So this is this line. But if I put just the right amount, I can control how quickly this clustering can take place. And so this orange line, you can see it never reaches this height, and it never really starts coming down like these ones do, because I'm not getting coalescence. So this basically says, by sprinkling in a few strong particles, stronger binding particles, I can drive this process more gently. So I'm kind of distributing nucleation over longer times. And again, I'm inhibiting gelation. And so I can control my crystallization outcome by seasoning, if you will. And in this case, I don't have a Gaussian distribution of interactions. I'm literally just using a second component. It's like adding a little bit of hot pepper to the system, but not too much, just to kind of spike it. So let me just leave it here in the interest of time. I'm just going to put these up, and just, the key point I want to make is that, these are two phenomena that are both really non-equilibrium in nature. Hydrodynamics does not affect the equilibrium outcome. It only affects the non-equilibrium behavior of the system. Heterogeneity likewise. And these are both instances in which they look like subtle experimental things, but in fact, they have profound effects on the assembly outcome. And they haven't really been studied much at all. People have always known that if you have flow, you can alter assembly outcomes, but this is without flow. This is purely due to a stagnant fluid introducing correlations between particles. And so this kind of brings to the notion that you can't just do equilibrium phase diagrams for these materials. You really have to kind of think about it from a material science perspective, where you include kinetic effects in addition to the thermodynamic effects, if you want to actually build structures that you care about. So I'll leave it here. Thanks very much for your attention.
Yeah, thank you for the great talk. So any questions from the audience?
So if for any audience you have question, you can unmute yourself to ask our speaker directly. So I have I have a question about when you model the particle-particle interaction, you in your model you introduce the lubrication force, but as we know, for the form of lubrication force, we know how for solid particle interaction, we know what's the form. But if you have some surface structure, for example, you have DNA or other kind of surface structure, how do you know? So what kind of form of the lubrication force you can introduce?
Yeah, so we don't really we don't worry explicitly about kind of the DNA on the surface of the particles. Okay, so these are literally just smooth particles. So the way, okay, so MPCD naturally allows you to couple the fluctuations and the forces between the particles due to the solvent particles. Now, what's the way that we introduced short-range forces? So the old way we did it was we added an analytical force based on lubrication. But the problem is, first, that's approximate. Secondly, it actually we only accounted for squeezing forces and not for shearing forces. The way we do it now is we use each particle is not represented as a single particle, but we use discrete particles. Okay, and it turns out the more discrete particles, and we're just actually writing this up now, if you use a rather large number of discrete particles on the surface, it allows you to use a very fine MPCD grid. And that automatically allows you to capture the diffusion at very close range between surfaces. So the lubrication forces are naturally built into this. They do not account for the effects of the polymer brushes coming with each other. And this is, in fact, a good point, not just because they affect the fluid, but also because if you think about two particles that are bonded by DNA, there's a sliding friction. There's a rolling friction because you have to literally rip bonds and form new bonds. And some people argue that this is also contributing to the effect that we see with the hydrodynamics. And that may very well be true. So we also have to introduce sliding friction, not due to hydrodynamics, but due to the Velcro effect of having these particles. So yeah, that's an open question, not included in here.
Yeah, thanks. So this kind of interaction will also affect the self-assembling and kinematics process, right?
Absolutely.
Okay.
Absolutely. I expect it very much. There's a lot of interest in, so if you take two planes of particles and you slide one over the other, some motions are allowed because particles can roll over each other. But now if I take this and I put a third layer below, now when particles slide over each other, they're rotating. That rotation might be incompatible with the layer below. So now you have to ask, can you rotate or do you have to slide? And those have very different friction effects and very different kinematic effects as well. And so that's something we have to think about a little bit more. But I think the hydrodynamic part is already a big piece of all of this.
Okay, thank you.
Yeah.
If I may ask a question on the same subject also. So thanks very much for this very, very nice talk.
Oh, thank you.
I appreciate it quite much. I'm wondering what about lubrication. Was it important to consider multi-body lubrication interaction, or it's just pairwise?
So within the framework, so we don't actually explicitly, like I said, so in the original stuff that I'm showing you here, which was done a couple of years ago now, it was pairwise.
Okay.
But the method that we're using now, which is to refine these particles so that they're made out of composite particles, so that naturally includes many body. Because every particle's motion is coupled to all the solvent atoms in the system. So there's no intrinsic separation between pairwise and many body.
Okay. Yeah.
So if you do it using these discrete particle approaches, it's the correct way to do it because you're capturing both sliding and squeezing lubrication and many body effects as well. I will point out the short range nature of the separation between these particles. I would argue if you really wanted to break it down, it's largely going to be pairwise effectively. And not, so the long range effects, of course, are many body, but the short range ones are dominated by the pairwise part. Even though we're not assuming pairwise, I suspect it's mostly pairwise.
Yeah, but if the whole system is coupled once in an implicit way, it might have a different behavior than when the dynamics is just pairwise.
That's right. I agree. I agree.
Yeah. Thanks
Absolutely.
a lot.
Yeah, no, thank you.
Oh, hi, Dr. Sino. This is
hi.
Joanne from University at Buffalo. Thank you very much for the very interesting talk. So in fact, I have two questions about your work. So the first one is I'm very curious about your time duration for all the aggregation process. So you mentioned the particle size is more like in the range of 10 to 30 nanometer. So I'm wondering what is the time range like in your case?
Oh, actually, so no, these particles are actually more on the 1 micron scale.
Oh, micron scale, okay.
Yeah, these are micron scale. So the DNA interaction range is 30, 40 nanometers.
Mm-hmm.
So this is very much a short range interaction. So think two very big spheres with a short range interaction. So the Brownian dynamics timescale here is, if I remember, microseconds to milliseconds.
Okay.
And actually, no, it's longer than that. But it's entirely based on the particle size.
Okay, so it can. Mm-hmm. So the simulation time scale can also match the experimental time scale.
That's right. We don't. So the last things I showed, which were these were done at cooling rates of. I'm trying to remember. These were done all the way up to the second time scale. Okay, so now it's much easier to do this in BD than it is to do it with MPCD. So MPCD is still a little bit challenging, especially if we're resolving the short range interactions, which means we're doing these discrete particle case. And this is, we're actually using Hongdi Blue from Sharon Glasser's group at this point to do the MPCD calculations because that code's already been implemented on GPUs. So we haven't run it on GPUs yet, but we're planning to do that, which will hopefully allow us to do MPCD runs that are similar in time scale as the BD runs. But you're absolutely right. The MPCD runs are not quite at experimental time scale. The BD ones can be, but not the MPCD ones.
Yeah. Thank you very much for this
Yep.
information. So my second question, in fact, about the crystal like a growth pattern. So I noticed that most of your case that you've shown here grow like very isotropically along the three like a dimension. So I'm wondering if there might be any mechanism which can control the growth just along one dimension, like a fiber pattern instead of a bulk, like a 3D pattern.
That's a great question. So I haven't really given that too much thought. So I will say this, we've had a long-standing interest, but not actually done anything about it, to look at growth from surfaces. So rather than a wire, just to have a substrate, and in particular, looking at growth on surfaces that have some kind of undulation so that we can actually pattern things. But to be honest, I haven't really done much in that area at all. There's no reason why, as long as you have some way to introduce a constraint, like a surface, or even have, I will actually point out, we had a project, this was somewhat orthogonal to this, but I'll just very briefly mention it, where we had a microfluidic chamber, and we had a channel with a little patch that was functionalized with DNA strands on the patch. And then we would flow the particles in, and they would then adhere to that patch. And it was a model, interesting enough, this was inspired by a blood clotting problem. We're trying to mimic platelet adhesion onto collagen. And so in this case, we could make monolayers of these particles, assemble them. So anything that can be, this is kind of the nice thing about DNA technology, is that if you can functionalize a wire with DNA strands, presumably you could then use that as your platform for assembly, and then see what grows from that. That would be interesting to do. We haven't looked at that.
Yeah, thank you very much for the further insight. Really appreciate that.
Oh, by the way, for comment for the last question. So actually, if the interactions or the DNA strands in the surface is non-uniform, instead of in all the surface, if it's only on certain locations of the surface, then it's possible. Because I was working on the sickle cell disease. The interactions between the hemoglobin is non-uniform, because it's non-uniform interaction in the surface. Then it will form a fiber.
Okay, good point.
So this is now non-uniform on the surface of the particle.
Yes.
Yeah. Okay. So this kind of goes into the whole. So yeah, I mean, people have been trying to design these so-called patchy particles for a long time.
Exactly.
Right?
Yeah.
Exactly. Right. And then you can form all kinds of filamentous type of assembly if you have sufficiently. Yeah. So. But I think most of the focus, at least in this area, has been to look at particles where you can make tetrahedral patches so that you can grow diamonds, because everybody wants to grow diamond structures because of their photonic properties. But absolutely, especially if you put, for example, the key limit, if you put just patches on the poles, north and south, then you're automatically limiting your growth to filaments. Absolutely, yeah. So the only challenge there is it's easy to do in simulation, not so easy to do it experimentally. That's always the issue.
But I have one question. So, is it possible to consider explicitly the DNA strands on the surface?
Surely, and people do. I know some groups have done that. So Monica Olvera de la Cruz at Northwestern's group has done a lot of those calculations. as an example, and she's actually worked with Chad Merkin on this. My opinion, it doesn't buy you that much. Okay, so I think it buys you something if you're specifically interested in nanoparticles. At the micron scale, resolving the strands really buys you very, very little. And in fact, I would argue that even spending too much effort on getting this potential just right doesn't matter. And there's actually a very, very nice kind of piece of insight from Dan Frankel, who basically showed that the phase behavior of these systems, when you get into the very short range limit, doesn't matter so much on the precise shape of the potential anymore. And so you can kind of lump everything into a virial coefficient and use that as a proxy for everything. And whether you have a square well potential or a DNA potential or a short range Leonard-Jones potential, they pretty much give you the same generic behavior. And so I'd say, you know, going in the other direction, it might be even better to just, because these models for the DNA potentials are still pretty messy to get the potential right. You could generate 90% of the phenomena, or I should say, almost everything you see will be largely unchanged if you made the potential just a short range Leonard-Jones, like a high exponent Leonard-Jones or something like that.
I see.
It's pretty generic.
Got it. Thank you. Any other questions from the audience? No? If there are no questions, I think we can stop here. Thank you again, Professor Cesium.
Thank you very much
thanks.
for the invitation. So very nice experience. Thanks for the questions and thanks for the invite.
Thank you. Bye-bye.
All right.